Okay, so welcome to this next video in which we are discussing the RAS proteins. Okay, so we're just in the process of discussing how the enzyme HRas is going to be moved from the endoplasmic reticulum membrane into the plasma membrane. Okay, so we've discussed that in order for it to be moved from the ER membrane to the plasma membrane, it needs to be palmitoylated. Okay, and this is an S-palmitoylation that's going to occur. We're going to add a palmitoyl group onto the phyl group of a cysteine residue in the hypervariable region. Okay, now this process is going to be catalyzed by an enzyme. Okay, and the enzyme that is going to catalyze it is a member of the DHHC family of enzymes. Okay, and this family of enzymes are all palmitoyl transferases, so they're going to transfer palmitoyl groups, and they're specifically S-palmitoylation enzymes, so they're going to transfer palmitoyl groups onto phyl residues. Okay, well, phyl groups of the cysteine residues. Okay, now palmitoyl transferases are all members of a bigger family, which you'll often see these enzymes referred to as, which is the protein acyl transferases. Okay, so protein acyl transferases. And this larger family of enzymes is just contains all enzymes which basically transfer acyl groups onto proteins. Now there are two main acyl groups that can be transferred onto proteins. Palmitoyl groups are one of these acyl groups, but there is another group known as a myristoyl group that can also be transferred onto proteins. So all the enzymes which catalyze myristoylation will also be in this family of protein acyl transferases. And for short, the family of protein acyl transferases is abbreviated to PATs. Okay, P for protein, A for acyl, and T for transferases. Right, so within this family of DHHC palmitoyl transferases, which specifically transfer palmitoyl groups onto cysteine residues within the protein, okay, so they specifically catalyze S-palmitoylation, there are 23 members, okay. Now, the specific one that is going to palmitoylate RAS proteins is a member known as DHHC9, okay, which is present both within the membrane of the endoplasmic reticulum and also the membrane of the Golgi apparatus. Okay, so let me show it here. So I'm going to show its membrane spanning topology here. Okay, so basically it has 4 membrane spanning alpha helices, and then it has an important region here which is known as the cysteine rich domain, okay, and it has another 2 membrane spanning alpha helices here. So let's have the carboxylic acid terminus here and the amino terminus here. Now, this is the cytoplasmic side of the membrane. Okay, so remember, we're either talking about the endoplasmic reticulum membrane or the Golgi apparatus membrane. So one side will face into the cytoplasm and the other will face into the lumen of either the endoplasmic reticulum or the Golgi apparatus. So this is the luminal side over here. Okay, now this special region here is extremely important in the catalyzing of the transferring of the palmitoyl group onto the cysteine residues. Okay, and it's known as the cysteine rich domain. Okay, so CRD for short. And within the cysteine rich domain there is a specific portion which has the sequence DHHC, and this is why this is called the DHHC family of enzymes. They all have cysteine rich domains, and in the cysteine rich domains, which are always on the cytoplasmic side of whatever membrane they're in, they always have a conserved sequence of amino acids, which is this DHHC. So you need to know your single letter amino acid codes to understand this. So D is the single letter amino acid code for aspartic acid. Okay, H is the single letter amino acid code for histidine. Okay, and then finally the C is the single letter amino acid code for cysteine. So let me just show you the R groups of histidine and aspartic acid, which are two amino acids that we haven't seen at any point in this video previously. So we'll start off with histidine. So I'm not going to draw the full amino acid structure, I'm just going to show you the R group. Okay, so you have a methylene group, and then you have a structure known as an imidazole ring, which is a 5-membered ring consisting of 3 carbons and 2 nitrogens. Okay, and you have 2 double bonds, one here and one here, and this double bond here between the carbon and the nitrogen, this is the double bond after which the whole ring is named, because basically when you have a double bond between a carbon and a nitrogen atom, it's called an imide bond, and the whole ring is then called the imidazole ring. Okay, so that should have been A, imidazole ring. Okay, so now just saturate everything by putting off some hydrogens. There should be a hydrogen there and a hydrogen there, and that now is the R group of histidine amino acids. Okay, right, aspartic acid R groups are basically ethanoic acid. They're an acetic acid group. Okay, so a 2-carbon carboxylic acid group sticks off as the R group for aspartic acid. So you have an aspartic acid followed by 2 histidines followed by a cysteine, and that's a conserved little pattern, a little motif that you have in all of these cysteine-rich domains of all of the 23 different types of DHHC enzyme. Now the specific DHHC enzyme that's involved in adding palmitoyl groups onto RAS proteins is DHHC9. So, remember, at the moment, the HRas protein will be anchored in the ER membrane, so it will be very close to this DHHC9 enzyme. Okay, and what's now going to happen is this DHHC9 enzyme is going to catalyze the adding on of a palmitoyl group onto a cysteine residue within the hypervariable region of the HRas protein. And once the HRas protein has a palmitoyl group added onto its cysteine residue within its hypervariable region, the HRas protein will move from the membrane of the endoplasmic reticulum to the Golgi apparatus. So let me again draw the picture of the different intracellular organelles. Okay, so here's the nucleus, and I'll draw it smaller this time so that I can fit in more intracellular more of the other intracellular organelles. Okay, so here's our Golgi. Okay, and what's going to happen is this HRas protein is going to go from being in the membrane of the ER here, and it's going to move to being in the membrane of the Golgi, and it will gradually move down the Golgi basically to the end of the Golgi over here. Okay, so it will move over to here, and then it will go from the Golgi into the plasma membrane. Okay, so its pathway goes from the ER to what's known as the cis Golgi. Okay, so the portion of the Golgi that is very close to the ER is known as the cis Golgi, which means on the same side basically as the ER, and then the portion that's furthest away and closest to the plasma membrane is then called the trans Golgi. So what will then happen is the HRas protein will move along the cisternae of the Golgi apparatus from the cis Golgi eventually to the trans Golgi. Then it will transfer out of the trans Golgi into the plasma membrane, and then it will be on the cytoplasmic side of the plasma membrane. And the palmitic acid group and also the farnesyl group will be sort of anchored into the plasma membrane, and that will hold the HRas protein at the plasma membrane. Now, let's now turn our attention to the final 2 RAS proteins, which are KRas4A and NRas. Okay, so the final 2 then, NRas and KRas4A, and we can group these 2 together. Okay, so they're going to go through the same process as HRas basically. They're going to be in the endoplasmic reticulum membrane, and then they're going to have palmitic acid molecules stuck onto cysteine residues within their hypervariable regions by this enzyme DHHC9. Okay, so let me draw this. So here we have our plasma membrane. Sorry, not the plasma membrane, this is the ER membrane. Okay, and then we have our DHHC9 enzyme here, with its cysteine-rich domain here. Okay, so this is DHHC9, so I'll just put the carboxylic acid terminus of this parent protein and the amino terminus. So this is DHHC9, okay, and then the KRas 4A protein, or the NRas protein, will be anchored in the membrane of the ER by its farnesyl group. Okay, now what's going to happen is DHHC9 is going to add on another another lipid molecule onto this protein, and it's going to be a palmitoyl group. So you're then going to get another group here. So let me highlight these two in different colours. So here's the palmitoyl group that has just been added on, and here's the farnesyl group in pink here. Okay, I think I've just totally swapped around on the colours that I used previously. I think previously I used pink for the palmitoyl and then orange for the farnesyl, but never mind. One is a farnesyl and one's a palmitoyl. Now basically, to get these proteins to go to the plasma membrane, a single palmitoyl group isn't enough. Basically, you need more. Okay, so you either need to add on more palmitoyl groups, i.e. you need more hydrophobic molecules added on, okay, or you need to do what KRas 4b did, and you need basic motifs added on. So you need basic molecules added on. So remember, KRas 4b have those 6 lysine residues, and all of those could accept protons, and when they accept protons they become positively charged. Okay, so if you had a basic molecule attached to you, then it could accept protons and you become positively charged, and this would help you go to the plasma membrane where you can bind to the phospholipids basically. So in order to get NRas and KRas 4A to move to the plasma membrane, they need additional groups adding on, either more palmitoyl groups or potentially some sort of basic group that can give the molecule a positive charge. Then what will happen is it will again move from the ER to the cis Golgi. It'll work its way down the cisternae of the Golgi apparatus to the trans Golgi. Then it will move from the trans Golgi into the plasma membrane, and then it's at the plasma membrane. Now, let's now discuss the depalmitoylation. Okay, and this only applies for those RAS proteins which were palmitoylated. So depalmitoylation. Okay, so remember, KRas 4b was not palmitoylated. It had that polybasic domain, basically, where it had the 6 lysine residues, which were all consecutive in the hypervariable region. And those gained protons and therefore gained a positive charge. And that helped to associate the KRas 4b protein at the plasma membrane. So it only needed the farnesyl group and those positively charged lysine residues to keep it at the plasma membrane. But remember, I did mention that it wasn't a strong interaction, so KRas 4b was always coming out of the plasma membrane and could go back to the Golgi membrane or go to the ER membrane and then potentially go back to the plasma membrane. Now, we're going to see a similar cycling, i.e. not always staying at the plasma membrane, but cycling between different membranes within the cell, basically. We're going to see a similar cycling for HRas, NRas, and KRas. So, let's now look at all of these. So the feature that all of them have is that they're held in the plasma membrane not just by their farnesyl group but also by palmitoyl groups, and they may have multiple of these palmitoyl groups. So remember, HRas will definitely only have one, but NRas and KRas 4A may have more, basically. Okay, but for our purposes we'll just draw it as one. Now basically, if you were to chop off this palmitoyl group, then the RAS protein, which is either NRas, HRas, or KRas 4A. And remember, we're not talking about KRas 4B now because it won't have palmitoyl groups stuck onto it. But if you chop off that palmitoyl group, then it will come out of the plasma membrane. And basically, it ends up going back to the Golgi. Okay, so which ends enzymes can do this? Well, basically there is an enzyme which is in the cytoplasm of the cell, and I'll draw it as a typical enzyme structure here, which is known as acyl protein thioesterase. Okay, so this is acyl protein thioesterase, and it's specifically acyl protein thioesterase 1. Okay, and this is often abbreviated to A for acyl, P for protein, T for thioesterase, and then 1. And this acyl protein thioesterase 1 enzyme can cleave off the palmitoyl group on the RAS protein, basically. And when it does that, the RAS protein will then only have the farnesyl group still anchoring it in the plasma membrane. And this releases the RAS protein from the plasma membrane, and basically it goes back to the Golgi apparatus, implants into the membrane of the Golgi apparatus, and there you will also have this enzyme DHHC9. Remember I said it was both in the ER membrane and also the Golgi membrane, and DHHC9 can then repalmitylate the RAS protein, and it can then move back into the plasma membrane, so you get this cycling. And this may be very important in regulating the RAS proteins, basically, because they're important in this signaling pathway. And I should have actually said probably right at the beginning what these RAS proteins are. So basically RAS proteins are monomeric G proteins, okay, which means that they have two states. They have an on state and an off state, as it were. Now in the on state they have guanosine triphosphate bound to them, okay, or GTP. So let me show this. So in the on state, a RAS protein will have the GTP molecule bound to it, and this doesn't just go for RAS, it goes for all G proteins. Okay, so this is the on state, and in the off state, RAS proteins will have GDP bound to them. So here is the RAS protein, and it has GDP bound to it. Okay, so this is the off state. So all G proteins have these two states. They have an on state and an off state. Okay, and basically there are certain receptors which can activate cascades, which will activate the conversion of the off state of the RAS G protein into the on state. Okay, and this will then trigger downstream pathways. Now, these signaling cascades that convert the RAS protein from the off state into the on state, they are all at the plasma membrane. So it's very important that RAS is, bound to the inner leaflet of the phospholipid bilayer and is nearby the signaling cascade that will turn it from the off state into the on state. Okay, and then it can activate the downstream pathway of it. Okay, now if the RAS was moved from the plasma membrane by breaking off these palmitoyl groups, which would take it back to the Golgi, then it wouldn't be able to participate in the pathways. So potentially this could be a mechanism for desensitizing the pathways, and this is of great interest because, as I say, the pathways that RAS is involved in generally activate growth. Okay, so they activate differentiation of cells and then proliferation of cells, and they're often mutated in cancer and they're overactive generally. Okay, so potentially being able to desensitize, deactivate these pathways would be very useful in cancer treatment. Okay, so one pharmacological correlate of this is that there is an inhibitor of this enzyme acyl protein thioesterase. Now this would not be useful at all in treating cancer, it would do quite the opposite. Okay, and if anything you want to activate this enzyme to reduce the expression of the RAS protein at the plasma membrane. Okay, but there is a pharmacological blocker of this, and it's known as palmostatin B. Okay, so this is palmo for palmitic acid, and then statin for keeping it static, B, so stopping the palmitic acid cycle basically of being added on and then cleaved off, added on and cleaved off. Okay, so that's a pharmacological agent that can be used to block the acyl protein thioesterase 1. Okay, so that now concludes our discussion of the RAS proteins. Okay, we've seen the 4 different types, and we've seen how they move from the synthesis of them in the cytoplasm to their presence in the plasma membrane, and the involvement of palmitoylation and farnesylation in this process.